Okay, so we get into our next kind of component where we're going to get into basically how things get in and out of the cell. And this is going to be really important as we go through the rest of the semester, because a lot of things that we're going to talk about, like the endocrine system and or the kidneys and all that, are going to involve things passing through cells, passing through membranes, passing through, right? So as we talk about things like muscle contraction, as we talk about how nerves conduct impulses, right? All sorts of things are going to involve cellular transport. And so we really want to get a firm grasp on this concept, since it's so pivotal in a lot of the different things we're going to talk about later on. So we're going to start by talking about the environment we find outside of the cell, the extracellular environment. We'll compare the intracellular environment and the extracellular environment, or ICF and ECF for short. We'll talk about the extracellular environment in terms of how it maintains normal cell function, the different fluids we find, and then the extracellular matrix, or the ECM. We'll talk about its function and what it's made of. So the intracellular environment is going to include the intracellular fluid, right? Whereas the extracellular environment has the extracellular fluid, and that's going to include things like plasma. It's going to include things like lymphatic fluid and interstitial fluid. Hopefully some of these sound familiar. Maybe you don't remember exactly what they are, but remember, plasma is what we find in the blood, right? It's the liquid part of the blood. Lymphatic fluid, right, is going to be found in the lymphatic vessels. And then interstitial fluid, this is the fluid we find in between the cells in the tissues. So we basically are looking at the different fluids we find. When we're talking about extracellular fluid, we're talking about the fluid within the bloodstream, within the lymphatic system, and then in between the tissues. So there's an important interaction, right, that occurs between the intracellular environment and the extracellular environment. They're going to influence each other. And so there's a necessity for communication, and therefore we have to then, because they interact with each other and they impact each other so heavily, it's very important that the composition of both of these fluids is regulated. If we start messing with the components, messing with things like the pH or solute concentration, right, so on and so forth, if we start messing around with that, we are going to impact both sides, right, the inside and the outside of the cell. And that can affect things like cells being able to do their jobs. So if we think about the interactions that happen between the inside and outside fluids with the cells, cells are going to receive nourishment from the extracellular environment. So think about like the bloodstream carrying around things like nutrients from the foods we eat, or oxygen from the air we breathe, right? That's going to be carried in that extracellular environment, and then is going to diffuse across for those cells to absorb, right, to pull in. The cells are also going to release waste products, and the extracellular environment is going to help get rid of those waste products so it doesn't build up, right? We don't want a pile of garbage, right? Just like you wouldn't want a pile of garbage building up outside of your house, right? You have trash collection that comes around. Same thing, the extracellular environment is going to handle waste products to prevent that buildup. And then we can use the extracellular environment to send messages. So cells can communicate by secreting chemicals. Some of these are going to be like paracrine, where it's very close, like we're just talking with our neighbors, right? Our neighboring cells. Others, like hormones, are going to be more systemic, that are going to be spread throughout the entire body. And the extracellular environment is going to be the side that's going to help move those chemicals from one place to another. Now, in that extracellular environment, it's not just a bunch of fluid. There's other substances found there as well, and that's what we call the extracellular matrix, or the ECM for short. I'm going to just call it the ECM from now on in this video, so if you hear that, just keep that in mind. So what is the ECM for? It's going to help support cells, right? So physical support, right? Think like scaffolding and whatnot. It's also going to help us segregate tissues from one another, so we have some organizing that's going on. It's going to help regulate intercellular communication, so regulating the communication between different cells. and also help regulate growth factors so that we can make sure that we're making more cells when we need them, but also not making them when we're just fine. There's a lot of different proteins, glycoproteins, and the ground substance that make up this ECM. So they're going to help with forming the tissues properly, right? So again, giving us some sort of structure, a scaffolding to build off of. Wound healing, right? So things like collagen, for example, will be pumped out in higher numbers when you have a wound, right? This is actually the basis for things like microneedling. So people who use microneedling to kind of bring back youth to their skin, right? What are they doing? They're creating all these little tiny micro wounds to stimulate that collagen production in the ECM. Fibrosis, scarring. So this is more what you probably notice this more when you have a wound that is maybe a little bit bigger, less superficial, where you see a distortion in how the tissue appears after healing from a wound. That is because there is more fibers, right, like collagen and whatnot, that are being pumped into that area and can cause scarring. It also plays a role in the metastasis of cancers. There's a laundry list of things that the ECM is responsible for, right? A lot of different things. But the main things that I want you to kind of remember is the structural support, organizing of tissues, and regulation of communication and growth. So let's go ahead and talk about some of these different things we find. Now that we've kind of gone over the overview, let's get into the nitty-gritty. So we have these protein fibers that are strung out throughout the ECM, as well as a gel-like ground substance that everything is suspended in. Some of those protein fibers include things like collagens and elastin for structural support. I mentioned those from the last slide. And then the ground substance, the jelly-like stuff, is going to be glycoproteins and proteoglycans, right? So glycoproteins and proteoglycans will make up that ground substance. Proteoglycans like fibronectin are basically going to extend from the cell cytoskeleton to anchor cells in place. So let's take a look at how this works. So here we have the microfilaments found in the cytoskeleton. So this is inside the cell, all these guys, and they're connected to this protein called integrin, to maintain the integrity of the cell, right? And then we can connect the cell by this integrin protein, attach it to the fibronectin proteoglycan, right? So we're basically anchoring the cell in place so that it doesn't stray from its current location, right? Obviously, it doesn't mean like it's held in place completely static with absolutely no movement, but it's holding it in, it's anchoring it down so that it can't just, in the fluid, in that extracellular fluid, just float away. And you may be wondering too, glycoprotein, proteoglycan, they sound really familiar, or I should say really similar. What's the difference between the two? So a glycoprotein is kind of generic. It's a protein that has a carbohydrate group attached to it, right? So kind of generic. A proteoglycan is going to be a protein with a specific group called a glycosaminoglycan group. And so that's the main difference between those two. So they're really similar, just proteoglycan is a bit more specific, glycoprotein is much more general. Now when we're looking at these things, when we're looking at the ECM, there's a net negative charge. This net negative charge is going to attract sodium ions, since sodium ions are positive. Sodium ions are going to attract water molecules because if we have that higher sodium concentration in an area, water is going to be drawn to it. And so this is going to keep the ECM hydrated so that we don't lose water and keep things fluid-like, right, gel-like, so they don't dry out. So let's go ahead and kind of look at some of the things we see in the skin for our example, right? So let's look at the ECM in the skin. So we know the very top layer, and hopefully some of these are a little bit familiar from anatomy, but if not, take a little time to look at that quickly. But we have the epidermis and the dermis and the subcutaneous tissues. In that epidermis, We're not going to get into the different layers. We have the keratinocytes that are producing keratin, that are getting filled with keratin, so that as they go up towards the surface, right, we provide that waterproof barrier. We have the melanocytes in there as well in the basement membrane, but we really see a lot of the cool stuff for the ECM down in the dermis. Remember that dermis is going to, at least for the reticular layer, is going to have that dense irregular connective tissue. And so remember connective tissue, we talked about ground substance and different fibers and whatnot that are in there in the fibroblasts. So if you don't remember that, try to maybe do a quick little review of it. But let's talk about the specifics of the ECM here and kind of relate the ECM and the different components of the ECM in general to this example that we've already seen in the past. So in the dermis, like I said, a regular dense connective tissue. Our ground substance in this case is hyaluronic acid. Hyaluronic acid is going to help attract water. That's one of the reasons why you see that component listed in skincare products when talking about hydration and staying hydrated. And so we have a hyaluronic acid ground substance, right? And this is going to be basically the gel-like substance, right, that everything is suspended in. Side note as well, it's also the basis of a lot of fillers. The fibroblasts are going to be the cells suspended in the dermis. These fibroblasts, if we think of the name, fibro, we think fiber, blast is going to be a cell that builds something. So a fibroblast is a cell that builds fibers. And namely, we're looking at two. We're looking at elastin and collagen. And there's different classes of like collagens and whatnot. We're not getting into that right now. But elastin, that's going to be for elasticity, suppleness, right? So when you stretch your skin and move it around, right, elastin is helping give that ability to stretch and move, but also go back to its original shape. And then collagen, this is for strength and firmness. It's a much more rope-like, stronger looking fiber compared to elastin. And so this is going to give us, basically kind of give yourself, your skin, shape, right? So as you get older, right, collagen production goes down, and so that's when you see things like wrinkles and things like your face just kind of seeming like it's sinking in, or that it is drooping a little bit more than it did in your youth. That's why.